Okay, good evening. So we'll begin the class now. Um, yeah. So again, today, since this will be an extra session, we'll cover questions from uh, all the different topics that were covered during the course. First is about biosafety level. So biosafety level dash equipment is required to work with the Ebola virus, and four different options are given here. You can take a couple of minutes to think about this, and then we'll go to the right answer. Okay, so if you see, this is how the biosafety level cabinets look like. For class 1, it's very simple, and class 2 is slightly advanced, except that you have a exhaust that's connected to this directly. In class 3, in addition to that, you also have these black colored gloves through which you can uh, hold and work with the culture. And here in class 4, you have these PPE suits. You have these PPE suits with which people work to avoid contamination. The second question is trypsin has an optimal pH of dash 1.5, 3.7, 7.4, 9.2. So, okay, so trypsin is an enzyme that is set to function. So, enzymes have something called as an optimal pH where they function, and this is how it looks like. Pepsin, being a digestive enzyme, works in acidic environment, so the optimal pH is between 2 to 4. But trypsin is produced in the pancreas, and it's cleaved in the small intestine to form trypsin. Now it functions maximally at pH 7 to 8. Fine. The next question is Dash is an initiator of acrylamide polymerization. Four options are given here: ammonium persulfate, TEMED, HCl, acrylamide. APS, that's what you've given. Yes. So here if you see in ammonium persulfate, your bisulfate radicals, this will react with your acrylamide monomer or your bisacrylamide. Now the point is this bis is what is used in cross-linking your acrylamide chains. Acrylamide forms the polymer and bis will cross-link these two. That is what gives you the pores. Now this is how uh, and here the bisulfate radical is needed so that it can connect your acrylamide monomers to form the acrylamide polymer. That is what is represented here. Fine. The next is if the amount of cross-linking reaction is more for polyacrylamide gel, the pore size will be dash. Smaller, larger, unchanged, no change in pore size, but the number of pores will decrease. Okay, so like we saw earlier, in your polyacrylamide gel, cross-linking is happening because it's a bisacrylamide, and if cross-linking is more, the size of the pore will reduce. So one thing that you need to know is your pore size and the number of pores are inversely related. If your cross-linking is very less, the number of pores will be more. Oh, sorry, if the cross-linking is less, the number of pores will be less. If the cross-linking is more, the number of pores will be more. So the right option is, as the cross-linking is more, the number of the pore size will be less. Okay. The next question is, the most appropriate technique to study DNA protein interaction is dash. Hydrophobic interaction chromatography, gel mobility shift assay, STS page, Western blotting. Hmm. Gel mobility shift assay. Yeah, that's because in gel mobility shift assay, what we see is we run the individual samples of DNA, then DNA plus protein, and then because here we run an agarose gel, we stain it with ETBR, which stains only your DNA. So when you see it, because the protein is heavy for the same electric field, um, because the protein bound DNA is heavy for the same electric field, there will be differences in the uh, mobility. And that is what we visualize as this distance or the shift. So it's gel mobility shift assay. The next is, which of the following techniques is used to find genomic binding sites of a known protein? Gel mobility shift assay, native horizontal page, DNA footprinting, 2D electrophoresis. Uh, you can take a couple of minutes to think about this. So, to find the genomic binding sites for a known protein, which of the following techniques is used? As we saw earlier, gel mobility shift assay will tell you whether DNA binds to the protein or the protein binds to the DNA. 2D electrophoresis is a method where we separate proteins based on two properties. One is their PI, which is the isoelectric point. The other is in the second dimension based on their molecular weight. So this way you get a distribution of proteins that are present within a given sample based on PI and molecular weight. So we've covered horizontal page. It's another method where pages run horizontally, but again because it's native, it's equivalent to that of a gel mobility shift assay. But DNA fluid printing is the method where the protein that binds to the DNA is then um, allowed to undergo uh, lysis, nuclease treatment. So the DNA that is not protected is identified and then sequenced as the DNA that is binding to the protein. The next question is, ion exchange chromatography separates analytes based on their charge, size, hydrophobicity, and molecular weight. Yes. So in ion exchange chromatography, the charge of the uh, analyte is used. In size exclusion chromatography, size is the size or the molecular weight of the protein is the one based on which separation happens. In hydrophobic induction chromatography, hydrophobicity is the one that is used. Molecular weight again is in SEC. So typically we use anion and cation exchanges, and this depends on the isoelectric point. So if the isoelectric point of the protein is around 5 to 6, what we do is we set the buffer pH to 8 and then use anion exchanges where your protein is negatively charged, and by eluting with NaCl you can get it. If your isoelectric point is around 8 or so, then what we do is we use cation exchanges. So typically DNA binding proteins have, for example, your polymerases or helicases, they have very high PIs, and in those cases we use cation exchanges. Yes, so in those cases we use cation exchanges. Uh, and when we use cation exchanges, again NaCl can be used, where instead of your chloride ion like in your anion exchanges, your sodium ion competes and helps. Dash helps in the binding of protein to a matrix in hydrophobic interaction chromatography. You can take a couple of minutes to think about this and then we'll discuss. Okay, yeah. So, if you see when the protein is present and in hydrophobic interaction chromatography, what happens? Your ligand is hydrophobic, and at different salt concentrations, different parts of your protein is exposed, and this can be typically altered. So, uh, at at higher salt concentration, this can bind, and at lower salt concentration, it is withdrawn. So what you typically need to know is at higher salt concentration, it increases. This enhances the hydrophobic interactions between the protein and the hydrophobic matrix. And this is based on the salting out effect. So the high salt concentration that reduces the solubility of the hydrophobic regions of the proteins in the aqueous phase, forcing them to interact with the hydrophobic ligand on the matrix. And at high salt concentration, structure of water is affected, making hydrophobic interactions more favorable. Elution. So proteins are here eluted by decreasing the salt concentration, which reduces the hydrophobic interactions and allows proteins to be released from the matrix. So salt concentration, changing of salt concentration is what will help in uh, eluting. So to reverse the elute, which is bind, you need to uh, increase the salt concentration. Again, a question along the same lines. Salting in is the process by which a high amount of salt increases solubility of protein. High amount of salt inducing protein precipitation, low amount of salt exposing a hydrophobic group of proteins, a low amount of salt increases the solubility of protein. You can take a couple of minutes to think about this and then we'll discuss. Okay, so salting is the process where we want to ensure the protein solubility is increased. So here what we do is we reduce the salt concentration, thereby ensuring the solubility is increased. So this typically follows a curve. That's what you see here. As you increase the salt, as you slowly reduce the salt concentration, you increase the solubility, and after a point, as you increase, your proteins will precipitate. So this is because of how the hydration shell is and how your salt interacts with the waters. The next question is: if a protein is incubated with 8 molar urea, for a long period of time, it will be eluted with dash. Four options are given here: Vg, Vo, Vi, and Vp. These are all different volumes. You can take a couple of minutes to think about this, and then we'll discuss. Okay, so when you incubate a protein with 8 molar urea, what happens is your protein gets denatured, all your non-covalent interactions is lost. So when your protein loses its non-covalent interactions, now it's more or less like a flexible, it loses its secondary structure and tertiary structure. So it will unfold into an extended random coil, and this will behave like a smaller molecule that can penetrate the pore fully. So they'll elute at the total column volume. The next is in irreversible affinity chromatography, the ligand receptor pair interacts with dash, covalent bonding, hydrogen bonding, van der Waals interaction, hydrophobic interaction. So this is irreversible affinity chromatography. Take a couple of minutes to think about this, and then we'll discuss. Yes. Yeah. So Sai Sampath has mentioned that it's covalent bonding. Yes. So covalent bonds are those that physically are present, like your non-covalent interactions. All your three other options, hydrogen bonding, van der Waals interaction, and hydrophobic interaction, are all non-covalent. So that's it. Next, to identify the peptide sequence of a protein after affinity purification, dash is used. Mass spectrometry, UV spectroscopy, fluorescence spectroscopy, cellular diffraction. So to get the sequence, what we use is mass spectrometry. So what we do is we digest the protein with proteases. Then these are separated and ionized, and then you get the mass. And then uh, those are again broken down. And based on this time of flight, and you get an idea of the sequence, and you match it. And so using mass spectrometry, you get the peptide sequence of the protein. Dash cells differentiate to form plasma cells, macrophages, B cells, T cells, dendritic cells. Yes, so B cells. So you have two different types of this activation. One is T cell dependent, the other is T cell independent. In T cell dependent, what you get is this antigen, which is the pink in color. These are uh, accepted by T cells, and the MHC2 presents it to B cell. And then you have either plasma B cells that are produced, or memory B cells. And here your plasma B cells are the ones that produce the antibody. In T cell independent activation, these memory B cells will undergo clonal expansion to produce plasma B cells that produces IgM, which is this pentameric antibody. So in short, when B cells encounter an antigen along with T helper cell signals, they get activated. And these can differentiate into plasma cells and memory B cells. So the plasma cells secrete large amounts of antibodies, and memory B cells provide long-term immunity. Macrophages, T cells, and dendritic cells, they do not differentiate into plasma cells. The next question is, monoclonal antibody is generated by DASH, hybridoma technique, purification of antibodies from the serum, STS page, Western blot. Take a couple of minutes to think about this, and then we'll discuss. In hybridoma technology, what we do is we immunize the mice with an antigen, and then what we do is we take the myeloma cells and we fuse it with the B cells. This gives us the hybridoma cells. Then we perform a selection where we screen the hybridoma for our antibody of interest by its ability to bind to the antigen. Once we get that, then we expand the hybridoma and get the monoclonal antibodies. The next is DASH is an example of immunodeficient test. Water-Loney method, ELISA, Western blotting, Northern blotting. Take a couple of minutes to think about this, and then we'll discuss. So for immunodeficient, uh, the major concept is that we have our antibody, then antigen, and because these are specific, we observe that it forms these curves, and this is due to diffusion. So you have three different cases where if it forms an arc like this with these two antigens and one antibody, then you get an identity. If you don't get a full arc but you get a spur, then it's called partial identity. And if it intersects, it's non-identity. The next is the bacterial dash is made up of peptidoglycan, plastids, cell wall, cell membrane, and nucleus. So in bacteria in general can be divided into two, gram-positive and gram-negative. Gram-positive bacteria have a thick cell wall that's made up of peptidoglycan. Gram-negative bacteria has a cell wall that is made up of an outer membrane and a peptidoglycan, where the peptidoglycan is thin and it is sandwiched between the outer membrane and the plasma membrane. So the one that is made up of peptidoglycan here is cell wall. Okay, cell wall. And one more thing, the other option, which is plastids. These are found in plant cells, not bacteria. Cell membrane, it's a bilipid layer and it doesn't contain peptidoglycan. And nucleus is absent in bacteria. Next question is, ribosomes are pelleted down at dash 300 G, 3000 G, 30,000 G, 3 lakh G. So ribosome is a protein complex that also contains RNA. Now this uh, has higher molecular weight. It's a very large machinery that synthesizes your proteins. Now, one logic that you can always keep in your mind is the higher the mass, the lower the RPM or RCF in this case, because it gives G is needed. So, for ribosomes, what they do is they do a method called as differential simplification. Here, first, the ribosomes that are smaller compared to nuclei and mitochondria, so they need more speed than nuclei and mitochondria. So, what they do is they centrifuge and then throw the pellet, take the supernatant, centrifuge it again, throw the pellet. Pellet is the one that is settling at the bottom. And they do this repeatedly, and specifically for ribosomes, these are pelleted at around 30,000 G. The next question is, in a cesium chloride density gradient centrifugation, the order of gradient components is dash. So in cesium chloride density gradient centrifugation, the molecules and particles are separate based on their Boyne density, and in general they are it's like how when you mix different solvents, petrol, methanol, water, they all arrange themselves based on density. So the one that is the lightest with the lowest density is on the top, the heaviest is in the bottom. Similarly, organelles that are the least dense, they settle in the bottom. Proteins that are immediate density, intermediate density, that are above organelles. RNA is more dense than proteins, so they'll be below proteins. DNA is the most dense. So yeah, and this is how typically it looks like: small sized particles, medium sized particles, and large sized particles as you finish the centrifugation. The next is the illumination of a specimen as well as the collection of light is achieved by a single lens and dash. Bright field microscopy, phase contrast microscopy, epifluorescence microscopy, dark field microscopy. You can take a couple of minutes to think about this and then we'll discuss. how the light goes through the objective and the lens. So in bright field microscopy, the illumination comes from below the specimen through the condenser lens, while the objective lens only collects the transmitted light. It's below, and the illumination and collection are not to the same lens. So the sample gets illuminated, then it absorbs light and it appears dark. It doesn't reflect light. If it reflects lights, then you'll be able to see the sample. But because the background doesn't interact with the light, the background is dark. So this is how it is. You have a collector, you have a condenser, you have the light source, and the sample is here. So it goes through this, and this is how we'll be able to view. Second is phase contrast microscopy. This is similar to bright field, and it uses transmitted light from below and modifies the phase difference in the light using a phase plate and the objective. Again here, illumination and collection are not through the same lens. So this phase shifter is what is used to modify the phase difference. So by phase, what we mean is uh, are the waves uh, traveling at the same? Is there any difference between the heights and troughs of the wave? Because this is where you have the concept called as interference that comes in, where if two peaks coincide, you get maximum, and if two peaks are opposite in the amplitude, then you get minimal or no, nothing visible there. So here, this is your phase shifter, and you have the objective. Here, your central beam is also blocked. The third is epifluorescence microscopy. So in epifluorescence microscopy, what we have is your excited light that is directed on the objective lens. Here, it hits the specimen, it excites the fluorophore. The emitted light comes back through the same objective. So this is how the it goes, and then it's connected. So in epifluorescence, we have a dichroic mirror that separates excitation and emission paths while still using the same. This is your dichroic mirror. Finally, dark field microscopy. Here, the illumination comes in from the side using the special condenser so that only scattered light enters the objective. The objective lens collects scattered light only, not the direct illumination. And here we can use uncolored and unstained samples as well. So the light from the background is excluded so that the background is dark and the sample is bright. This is your dark field microscopy, where your sample is bright and the background is dark. The next question is, to make a sample conductive, it is dash coated with a conductive material, coated with silica, coated with liquid nitrogen, coated with carbon nanoparticles. You can take a couple of minutes Right. So to make the sample conductor, what we do is we coat it with a conductive material. This is called as sputtering. So the thing is, in scanning electron microscopy, if the sample is not conductive, when the electron beam is hitting it, it will get charged up. So the image that you get is bad. It doesn't represent what is really there. So to prevent this, what we do is we coat it with a thin layer of conductive material of either gold or platinum or an alloy of gold and palladium, or in some cases carbon as well. Now silica, that's an insulator, so that would not help us with conductivity. Liquid nitrogen, it's used in cryosol. That is only for freezing, but not for conductivity. And carbon coating, it is used, but not nanoparticles. It's usually a thin conductive layer. The next question is the principle of amplification of DNA with a modified nucleotide used in sequencing of prime molecules by which of the following four methods? Sanger method, Maxim Gilbert method, Edmond degradation, mass spectroscopy. Take a couple of minutes to think about this and then we'll discuss. So, for sequencing of DNA, we can use dideoxynucleotide where chain termination will happen. So, in this method, what we do is we have a particular nucleotide that would not allow your chain to be terminated. So, we have these combinations. We have deoxy ATP, deoxy CTP, deoxy GTP, and deoxy TTP. With this, if you had one of your dideoxy NTPs, what would happen is it would terminate at different lengths. And if you run, let's say, each of these in a single cell, A, C, G, and B, with that you'll be able to get an idea about what is the sequence. The next question is cleanout fragment lacks dash polymerase activity, 5' to 3' exonuclease activity, 3' to 5' exonuclease activity, proofreading activity. You can take a couple of minutes to think about this and then we'll discuss. Okay, so your DNA polymerase, uh, when it is cleaved by a protease, what will happen is the small fragment will be removed. This is involved in primer removal and it does this by having 5' to 3' exonuclease activity. So the other large fragment is involved in polymerase activity and 3' to 5' exonuclease activity, which is needed for proofreading. So your cleanup fragment it will lack your 5' to 3' exonuclease activity. It is needed for primer removal. The last question for today is protein sequencing by Edmond degradation is carried out by degrading the peptide from the dash C terminal, both C and N terminal, central amino acid of the peptide. Take a couple of minutes to think about this. We'll get back to this. Okay, so in Edmond degradation, what we do is we use phenyl isothiocyanate and that will attach to your end term, and then we can degrade it with the in under acidic conditions. And then when we run it on THC or HPLC, based on the retention, we'll get to know what amino acid this is. So this is how proteins can be sequenced using Edmond degradation method. So with this, we come to an end for uh, the extra session for NPTEL experimental biotechnology. If there are any questions, you can let me know. Thank you.